Clover Hill. This is Johnson Biology News Network. Tonight, our top story, a diamond in the rough. A new species found in the Great Basin of Nevada, Buffo williamsi. The Great Basin is a very arid place, and there's not a lot of water, so it would be kind of uncommon to find a new species of amphibian in this kind of place. The Great Basin at one time was a marshland during the Ice Age. It was extensively studied over a 190,000 square mile space by Richard Tracy. His study was 10 years long, and his evidence supports the possibility of 3 distinct species, but only one is determined as of now. The Dixie Valley toad, as it's normally called, is an olive-colored toad with small golden marks on its back. Found in Churchill County, about 100 miles east of Reno. A power plant is to be built in the area, but researchers are looking into that to see how it will affect the life of the new species. Tracy has received the American Society of Zoology Service Award and many other awards in his study. Back to you, Morgan.
Breaking news this afternoon. A new biological discovery involving fire ants has caught the attention of many people. Fire ant venom contains a toxin called solenopsins, similar to ceramides, which restore the barriers in the skin. During a 28-day experiment, scientists rubbed a skin cream containing fire ant venom on mice psoriasis models. The results are fantastic for those suffering with psoriasis and other autoimmune diseases. The mice treated with the skin cream had a decrease in skin thickness and inflammation. Additionally, the solenopsins have been found to restore the barriers in the skin, unlike the current steroids used to treat psoriasis. Back to you, McKenna.
Thanks, Morgan. Now, we shift our focus towards a recent study published September 6th that talks about the man's best friend. That's right, we're talking about dogs. In an effort to protect endangered carnivore species in Argentina, scientists are seeking the help of these furry companions to help track the movement of carnivores, such as jaguars, pumas, ocelots, oncillas, and bush dogs. The idea is that the service dogs were trained to sniff out a particular carnivore's scat, and then a DNA analysis was conducted. From there, the DNA analysis revealed that animals' movement and how movement was influenced by habitat quality, topography, roadways, and human disturbances. The conclusion? After combining 10 years of data for the endangered carnivore species into a corridor model, the study shows that all the carnivores overlap in their ecological requirements. Katie DiMatteo says, however, this is not without variation in the degree of habitat flexibility. The corridor model included a private properties which allows for communication and then a minimized cost. Now let's go back a little bit and ask ourselves, so what? Why should we care? Why can't we just keep them in one small place like before? Well, here's your answer. The 2 main reasons why a species becomes endangered are loss of habitat and lack of genetic variation. Who's at fault for this? None other than ourselves, humans. Human enrichment in the environment has always been detrimental to the survival of many animals. We make efforts to protect them in these small protected reserves. However, other recent studies show that they are not enough to ensure the survival of the species because species must move and breed to maintain genetic diversity. The goal of the study was to provide the optimal footprint for proposed habitat connection corridors and still be able to be flexible with the needs of the private landowners. And I believe they did just that. Sophie, back to you.
Thank you, McKenna. Now we will look at a team of researchers from the University of Texas and Stanford University School of Medicine. They used a cast-aside technique to begin determining the function of circular DNA and the connection to the human genome. They published their findings on September 11, 2017. Normal DNA, called chromosomal DNA, makes up our genes and chromosomes in our cells, and is in the shape of a double helix. However, extrachromosomal circular DNA is found in each cell's nucleus, and the function is still mostly unknown. It is in a circular shape with no loose ends. The team of researchers used density gradients centrifugation, a lab technique created 50 years ago, to separate DNA based on density because chromosomal DNA is less dense than circular DNA. The DNA is mixed with a dense salt solution containing cesium chloride in a small test tube along with dye that binds differently to both linear DNA and circular DNA. The higher density circular DNA concentrates in a band near the bottom of the tube, and modern techniques were also used to determine that a clean sample is collected. The researchers found circles that were copies of coding regions of chromosomal DNA that contains genes for making specific proteins. Circular DNA may be used as a potential diagnostic biomarker for certain pathogens like cancer. They are researching the circulome in healthy tissue and that in colon cancer tissue in the same person. Their hopes are to open up more information about the human genome. And now to our next segment with Nathan.
Thank you, Sophie. Our newest story on Johnson Biology News Network covers the discovery of how zebrafish obtain their unique stripes. Published last month by Dr. Woolley of Cardiff University, he discovered a key factor to how zebrafish generate their patterns. When zebrafish are born, they do not have an ingrained pattern already prepared for them in their DNA and live their early life as a transparent embryo before developing an iconic pattern. They contain 3 different colored pigment cells, which include black, yellow, and silver, which chase each other to form their patterns. While growing into adults and creating their pattern, many mutations may occur that dictate the pattern that develops in the zebrafish. These patterns can include distinctive stripes, broken stripes, polka dots, and sometimes no patterns at all. The patterns are created from the 3 different colored pigment cells chasing each other hundreds of times before yellow cells eventually push the black and silver cells to form the final pattern. Experimentalists found that when 2 types of these pigment cells are put into a petri dish, one cell would chase another in a spiral shape instead of the regular straight line. Dr. Woolley followed up the experiment by running computer simulations that took into a broad account of how the cells move and interact with one another in a zebrafish that are only a few weeks old. By experimenting with these different chasing angles, Dr. Woolley was able to replicate the different patterns that zebrafish have demonstrated. Now back to you, Deidre.
Next on Johnson's Biology News Network, we will address so-called smart molecules, which have been genetically engineered to fight against cancer. Bioengineering professors Peter Yingxiao Wang and Xu Qian, in collaboration with Victor Nizet and Jiangdong Zhu, call these smart molecules iSNAPs, also known as integrated sensing and activating proteins. And they have been created to override a cancer cell's ability to remain unrecognized and undisturbed in the immune system. The protein CD47 on cancer cells sends improper signals to macrophages, white blood cells, leaving them virtually invincible. Genetically engineered iSNAPs function as they do because they have been altered to interpret the signals which tell them to overlook the CD47 proteins as a signal to focus on them. iSNAPs have been essentially rewired. Furthermore, these iSNAPs can have a reaction time of only a few seconds. In the future, Wang hopes the principles they have put into action can be useful in the future by re-engineering other misguided cells to win against numerous other infectious diseases. Now on to other news.
Thank you, Deidre. Today on Johnson Biology News Network, we are closely watching the behavior of marine animals as a recent discovery on how plastic nanoparticles affect their behavior. The production of plastic is becoming a huge environmental concern for animals not only on land but also in the water. About 10% of world-produced plastic ends up in the oceans. There has been a recent study done to show how these tiny plastics end up in the brains of innocent fish. The size of the plastic particle does affect how damaging it is. Nanoparticle plastics are far more dangerous than large-sized plastic particles because they are able to cross the blood-brain barrier, thus leading to the brain and causing behavioral changes. It's important to note that the plastic nanoparticles cannot cross over to humans by consumption of the fish. These particles can only be transported by aquatic species. That's all for today. Back to you, Sophia.
Thanks, Allison. In a new initiative sponsored by the Indian government to test the DNA of every frog and toad species in the country, herpetologist Elizabeth Prandini reports that a new species of frog has been discovered. The purple frog has been discovered in the Western Ghats mountain range of India, which is home to numerous species of frogs. The new frog has been named in honor of famous herpetologist Dr. Subramanian Bhupathi, who passed away in 2014 while researching the Western Ghats. Currently, the frog's common name is Bhupathi's purple frog. This species has a mouth containing a long tongue designed to suck up food such as ants and termites, as well as small feet in the shape of spades. Herpetologists believe that these interesting characteristics are due to the frog's unique choice of habitat. The purple frogs spend the majority of their lives underground. and use the long tongue and spade-like feet to eat, eliminating the need for the frogs to surface. Although the frogs spend the majority of their lives underground, they begin their lives above ground during the summer monsoon season in India. First, the eggs are deposited in streams along the mountainside, and after a couple of days, the tadpoles hatch. These tadpoles are equipped with mouths that enable them to suction onto the rocks in the stream. The tadpoles make this their home for the next 4 months and eat the algae that lines the rocks. After 4 months, the tadpoles have become adult frogs that disappear underground for the majority of their lives. As a result of the frog's unique abilities, Bhupathi's purple frog has joined one other frog in its family, which is a relatively new family. Its scientific name is Nascapterygus bhupathi. This family contains 2 purple frogs in the western Ghats mountain range. The other species was discovered in 2003. The closest relatives to this frog live in Seychelles, and herpetologists are interested in learning more about the species. More information can be found in the scientific journal Elitis, where herpetologist Elizabeth Prindini published an article discussing the recent discovery. Back to you, Kimberly.
Thank you, Sophia. In a first, scientists at the Oregon Health and Science University used a molecule called CRISPR-Cas9 to alter the mutated human gene responsible for causing hypertrophic cardiomyopathy, a disease affecting the heart. This is a CRISPR molecule. It has 2 parts, an enzyme called Cas9 and a changeable RNA sequence that guides the molecule to the diseased DNA that matches the RNA. In the experiment, the researchers injected CRISPR into a healthy egg cell fertilized by a diseased sperm cell. In addition, they also added a DNA template to replace the mutated sequence. CRISPR works by going through the DNA sequence, searching for a match to its RNA. When it finds one, it uses molecular scissors to remove the mutated DNA. This leaves a hole that the researchers expected to be filled by the template DNA, but instead is replaced by the mother's healthy gene. This embryo self-editing came as a surprise and meant that it would be hard to fix mutations when both the parents have it. However, when repaired, the disease is not only removed from the subject, but also future generations. Sometimes the cell copies the DNA before CRISPR is able to correct it, and a mutant gene escapes, resulting in a mosaic embryo, which has some repaired DNA and some mutated DNA. Therefore, the disease could still be passed to new generations. Researchers found that by adding CRISPR before fertilization, mosaic embryos could be avoided. This diagram illustrates the differences between the 2 methods. The top method shows CRISPR being added after DNA replication and therefore producing mosaic embryos. As you can see, the sperm DNA is only partially repaired, whereas the bottom method depicts CRISPR being added with the sperm and only makes healthy embryos. It is important that all embryos are edited because if there is one mosaic, the whole process is ruined. These findings achieved the researchers' main purpose, which was to improve the safety and efficiency of gene editing for eventual clinical trials. Although implanting these embryos are far in the future, scientists can see CRISPR being used in big ways, such as battling HIV, eliminating Huntington's disease, and altering a mosquito's genome to prevent the spread of malaria. Back to you, Zaid.
Good evening, and thank you, Kimberly. Tonight, breaking news is scientist Anna Stinger discovered how plasmids and chromatophore are made out of the same proteins even though they evolved over 1 billion years apart. It all starts with the simple protist that is called a pollinella. This simple organism is host to chromatophore. Now, when it becomes time for pollinella to create chromatophore, it has a large number of proteins inside the cell that it creates to form the chromatophore. Then, Anna Stinger discovered the different proteins by using mass spectrometry. Mass spectrometry is when a scientist uses the different pigments inside of a protein to determine what proteins are inside of a cell. In addition, additional tests were used to determine the amount of long and short proteins put inside of the cell to determine the better quantifications of what proteins were used to make chromatophore. That is all the time we have for now. Back to you, Lainey.
Thank you, Zane, and welcome back to Johnson Biology News Network. I'm Lainey, and today we're going to be learning more about one of the world's largest animals, or should I say fish, the whale shark. The average size of a whale shark is 12.5 meters to 41.5 feet, which is roughly the size of a school bus. Even though whale sharks may seem intimidating because of their size, these massive animals only feed on tiny drifting animals like copepods or anchovies. Because of the enormous size of the shark and the tininess of the prey, whale sharks have to travel long distances to get the needed amount of food. Even though researchers know how much and how the whale sharks eat, their migratory patterns remain relatively unknown. Because scientists do not know the migratory patterns of the shark, biologists cannot protect the whale sharks from shark finning, so whale sharks ended up on the endangered species list in 2016. In August, biological oceanographer John Ryan and other teams of scientists satellite tagged 27 whale sharks from Darwin's Arch. Darwin's Arch is located near the Galapagos Islands and in the eastern tropical Pacific. This was the first time any whale sharks have been tagged in the eastern tropical Pacific, and the results were very insightful. The study showed that the sharks traveled east and west and stayed between the equator and 5 degrees north latitude. Ryan analyzed the migratory patterns of the sharks along with ocean temperatures in order to see if there was any correlation between these two pieces of information. After Ryan finished his analysis, he determined that the sharks were traveling in between cold and warm boundaries. The boundary that the sharks were following is called the North Pacific Equatorial Upwelling Front. And on both sides of this front, there are areas of large sea life. Basically, the whale sharks were traveling along a giant feeding highway. With this newfound knowledge, thanks to Ryan and his team, whale sharks are being protected by marine biologists, and within due time will hopefully be removed from the endangered species list. That's it from me. Now we will hear from Adaya, where she will talk to us about rodents.
Thanks, Lainey. Similarly, on the topic of animals, a story has surfaced of a brand new mammal discovered in the Solomon Islands. The new mammal is a giant tree rat, scientifically named Uromia spica, but known among locals as Bika. The new mammal looks like a mix between a squirrel and a possum rather than a rat. It is considered a giant tree rat because it weighs over 2 pounds and measures over 1.5 feet in length. The existence of the rat was not unknown. Many locals at Zaira, a village on Rangoonu Island, have passed down oral stories of the elusive rat, but it had yet to be found. So when scientists went to track down Bika, they saw many clues on the way that led them to their discovery. They found little holes extracted from nuts and coconuts on the island. Local rumors even stated that the rat could crack open coconuts with only its 2 front teeth. Researchers have identified 3 types of rats under the genus Uromes before the discovery of Bika. They believe that the rare mammals arrived to the Solomon Islands by rafting on vegetation from either New Guinea or Australia. Once they arrived on the islands, they evolved into a new species that adapted to their environment. The 3 species are the Uromes imperator, Uromes procellus, and Uromes rex, and now the newly discovered Uromia spica. The Uromes imperator and Uromes procellus have not been documented since their discovery. Many believe they have been driven to extinction by feral cats. To make sure the Uromes spica was a different species than the previous Uromes, many tests were done. Scientists compared cranial and dental morphology to the nearest millimeter, as well as molecular genetic sequence studies. Their guard hair of the Uromeus rex looks like this, compared to the guard hair of the Uromeus spica, which looks like this. Cranial dental characteristics of the Solomon Islands rodents and phylogenetic analyses confirmed the Rangoonu Island specimen represents a new species of the genus Uromes. Talk about a rat that likes to play hide and squeak. Now over to Ben for our news story.
Thanks, Adaya. Computational biologists at the Public Library of Science have recently published a new study on the effectiveness of 3 fluoroquinolones, moxifloxacin, levofloxacin, and gadifloxacin. Fluoroquinolones are parts of a multi-drug tuberculosis therapy. Biologists at PLOS have ran several computer models and simulations testing the various fluoroquinolones' effect on clusters of tuberculosis cells inside the lungs, called granulomas. In the end, scientists found that their data pointed to moxifloxacin as the best drug for the job. Not only was it most effective at reducing the size of the granulomas, but moxifloxacin was also found to perform fairly consistently regardless of the irregularity of simulating the patient missing doses. So even if the patient missed doses in the simulation, the drug performed pretty consistently. Figuring out which drug works the best will help future researchers eliminate variables in their experiments because they better understand how these drugs each work on their own. With the latest scoop, here's Ryan.
Thanks, Ben. A new test developed at the California Institute of Technology that identifies antibiotic-resistant bacteria in as little as 30 minutes could help turn the tide in the fight against superbacteria by allowing medical professionals to better choose which antibiotics to treat an infection with. When doctors treat patients with bacterial infections, they often skip over first-line antibiotics like methicillin or amoxicillin, drugs that bacteria are more likely to be resistant to, and go straight for stronger second-line antibiotics like ciprofloxacin. This practice increases the chance that the treatments will be effective, but the increased use of these makes it more likely that bacteria will become resistant to these stronger drugs. According to Nathan Shope, a co-author of this study, right now we're overprescribing, so we're seeing resistance much sooner than we should for a lot of antibiotics and that we would otherwise want to preserve for more serious situations. Prior to this study, there had not been a quick and easy way for doctors to know if their patient's infections is resistant to particular antibiotics. To find out, they would have to send a sample up to a testing lab and wait 2 or 3 days for an answer. According to Rustem Ismailov, therapies are driven by guidelines developed by organizations like WHO and the CDC without knowing what the patient actually has because the tests are so slow. We can change the world with a rapid test like this. We can change the way antibiotics are prescribed. Ismailov, Shope, and co-author Travis Schlappe aim to develop a test that could be completed during a single visit to the doctor's office. They focused on one of the most common types of infections in humans, UTIs, which 50% of women contract during their lifetimes. UTIs result in 8 million doctor visits and 1 million ER visits each year in the United States alone. The researchers' new test looks like this. A sample of urine collected from a patient with a UTI is divided into 2 parts. One part is exposed to an antibiotic for 15 minutes, while the other part incubates without antibiotics to serve as a control. The bacteria from each sample then are broken open, lysed, to release their cellular contents, which are run through a process that combines a detection chemistry technique called digital real-time loop-mediated isothermal amplification, or DLAMP for short, with a device called slip chips, which are a previous invention of Ismailov. This combination replicates specific DNA markers so that they can be imaged and individually counted as discrete fluorescent spots appearing on the chip, which would appear right there. When used on 54 samples of urine from patients with UTIs caused by the bacteria Escherichia coli, the test had a 95% match rate with those obtained with the standard two-day test, meeting the gold standard for accuracy and being a fantastic harbinger for what to expect in the future. In this future, the researchers look to other types of infectious bacteria to see how well it performs. They also hope to tweak the testing procedures to work with blood samples, as blood infections are more difficult to test since the bacteria are present in much lower numbers than in urine. However, such a test could reduce the mortality rate from blood-borne infections, which can turn fatal if not treated quickly. For more information, a paper describing the study appears on November 4th issue of the Science Translational Medicine. Back to you, Shri.
Thank you, Ryan. While TB has declined in the U.S. and other developed nations, the infectious lung bacteria remains throughout developing nations, where mutant versions of the disease become more and more resistant to current drug treatments. 2 studies led by the Translation Genomics Research Center and the University of California, San Francisco investigate a new genetic sequencing test that eliminate threats posed by a drug-resistant mutations of TB. In both studies, however, the TGen and UCSF use single molecule overlapping reads, also known as SMOR, technology to precisely identify variants of TB, even in cases where an older technology, also known as Sanger sequencing, found no drug-resistant mutations. A version of this SMR test is being developed to provide to underdeveloped areas where it may be most needed. The second TGen UCSF study, Mycobacterium TB subculture results in a loss of potentially clinically relevant heteroresistance. According to Dr. McAuliffe of TGen, he believes this further emphasizes the need for additional study of the effects of this phenomenon on both clinical diagnostics and patient outcomes. Identifying rapidly mutating drug-resistant strains of TB is the greatest challenge to eradicating this disease, which in 2015 infected 10.4 million and killed 1.8 million people across the globe, according to the U.S. Centers for Disease Control and the World Health Organization. Back to you, Kuba.
Thanks, Sri. Breaking news. Brain cells that control appetite have been identified. These cells, called tannocyte cells, were thought of as cells that respond solely to glucose. However, a new study suggests that they may also be responsible for detecting amino acids. Tannocyte cells are located in the hypothalamus, the region of the brain regulating hunger, in the third ventricle of the brain's ventricular system. Glucose sensing is dependent upon ATP receptors, and in some tannocytes, the sweet taste receptors TAS1R1 and TAS1R3. Closely linked to those receptors is the TAS1R2, located in the tongue, which detects umami taste, the flavor of amino acids. This suggests that tannocytes could detect amino acids via umami detectors. Researchers humanely killed rats, sliced their brains, kept the hypothalamus, cut it in half along the third ventricle, and exposed the tannocyte cells for study. Two amino acids that react with tannocyte cells are arginine and lysine, found in pork shoulder, beef sirloin steak, chicken, mackerel, plums, apricots, avocados, and almonds. At low concentrations, arginine and lysine were introduced to the cells, and within 30 seconds, information was sent further into the brain to let the organism know that it is full. A distinctive property of the TAS1R2, the amino acid taste bud, is the allosteric modulator inosine 5' monophosphate, IMP for short, enhances responses of umami taste buds. Furthermore, the researchers added IMP to arginine and found an increased amount of activity in tannocyte cells through fluorescent calcium detection. Research also suggests there are more ATP receptors within tannocytes that respond to amino acids. The MRS-2500 is a known receptor antagonist for glucose. However, it alone has no significant effect on the detection of amino acids. Only a combination of the P2X and MRS-2500 antagonists reduce the effectiveness of the receptors, indicating that there are more receptors available for mediating responses to amino acids. The larger amount of receptors also suggests that this is a physiologically important mechanism. Another piece of evidence supporting the significance of the mechanism is that if the TAS1R2 umami taste buds are lost, another receptor will compensate by detecting a range of amino acids and sending that information to the brain. Another way that tannocyte cells detect amino acids is through direct contact in the cerebrospinal fluid. Once amino acids are ingested, they go to the bloodstream. A portion of them crosses the blood-brain barrier and permeate into the hypothalamus. From there, the amino acids are detected. This direct detection, along with umami taste detection, gives plenty of opportunity for tannocyte cells to recognize amino acids in the body. Professor Nicholas Dale of Oregon University says, quote, amino acid levels in the blood and brain following a meal are a very important signal that imparts the sensation of feeling full. Finding that tannocytes located at the center of the brain region that controls body weight directly sense amino acids have very significant implication for coming up with new ways to help people control body weight within healthy bounds. Quote, if the tannocyte cells are activated without anything being put into the digestive system, the urge for food could be suppressed. The researchers' discovery has a proposal for the role of amino acids in neurogenesis, or formation of new neurons. P2Y receptor stimulation is required for neurogenesis of neural stem cells. P2Y also happens to be an ATP receptor for amino acids and glucose. Therefore, it may be possible that the intake of amino acids or glucose could trigger neurogenesis. Thank you for tuning in. Now back to you, Renee.
Thank you, Kuba. Now on to our next topic of discussion, cells. More specifically, how a team of researchers at the University of Warwick...
Wait, hold on, sorry. You need to be standing over here.
Oh, do I need to move over?
Yeah.
Dang it. Okay, start over. Thank you, Kuba. Now on to our next topic of discussion, cells. More specifically, how a team of researchers at the University of Warwick, located in Coventry, England,
have discovered a way to genetically engineer cells to fight disease. The study was led by Professor Alfonso Jaramillo, who discovered that RNA, also known as ribonucleic acid, can be sequenced to read code, similar to the way a computer or an app on our smartphones would. RNA is a common molecule produced generously by all living organisms and serves to transmit information between DNA and the proteins in a cell. Professor Jaramillo comments that this breakthrough would allow RNA to sense its environment and control gene expressions without relying on proteins for indicators as it normally would. This means the sequences would be applicable not only throughout the animal kingdom, but the plant kingdom as well. So, for example, plant cells could be genetically modified to be immune to certain pesticides or other environmental factors. As supported by the team's research, these RNA sequences can be created to allow cells to have what they call a virtual machine, which would act as a translator that enables cells to specifically understand coded instructions. These instructions could program the cells to attack certain diseases like the flu or even cancer, which would allow for healthcare plans so individual that it is literally tailored right down to the patient's cells. Now back to you, Caroline.
Thank you, Rachel. Now, news on the ever-raging debate of human superiority. Research published in August by experts at the University of Portsmouth say that decades' worth of information on apes' intelligence may be incorrect, as results have been shown to largely have been influenced by a bias towards humanity, and unfair tests based off of human traits. Therefore, apes may be even more intelligent than previously thought. Dr. David Levins of the University of Sussex worked together with Professor Kim Bard of University of Portsmouth and Professor Bill Hopkins of Georgia State University. Dr. Levins explains that the fault underlying decades of research and our understanding of apes' abilities is due to a strongly held belief in our own superiority, and that, as humans, we see ourselves as top of the evolutionary tree, which leads to biased research designs against apes. Dr. Levins goes on to explain how, even in experiments where apes clearly outperform human children, which have been praised as being much more capable than even adult apes, the ape's success is somehow still contributed to its inferior capabilities. In addition, Levins states that the researchers have demonstrated lack of understanding on the differences between apes and humans when it comes to interpreting and understanding body language and gestures, as there is not one scientifically sound paper published on the subject. Levins compared the faulty research and claims to those of experts of the past who deemed Northern Europeans to be the most intelligent race. Furthermore, Professor Bard points out the gap in literature between real evidence and opinion, how the common trend suggests a deeply rooted desire, even in academic groups, to name humankind as the only species capable of sophisticated social intelligence, an idea unsupported by the evidence. As an example, the action of pointing is brought up. If an ape makes a pointing gesture, the meaning is considered to be uncertain. However, if a human does the same, the assumption is that humans have a degree of sophistication brought about through the process of evolution, which other species clearly are incapable of possessing. To solve this problem, Professor Bard wonders if current comparative or developmental psychology could contribute to the field's overall understanding of the cognitive foundations of communication development. Bard considers this to be important, as he says, for researchers interested in the origins of language, focusing on behaviors without considering the animal's specific learning experiences will easily and inaccurately load results in favor of humans. Two main studies found to be guilty of this bias are brought up by Levins, Bard, and Hopkins. Firstly, they mention a set of experiments in which children, raised in a community full of the cultural conventions of nonverbal signaling, are tested against apes, raised without this cultural exposure, on their understanding of Western conventions of nonverbal communication. In this, of course, the children outperformed the apes on most tasks. In the second study mentioned, human children were compared to adult apes, and it was concluded that humans alone have been evolved to point towards an object that is not currently within view. However, this experiment failed to take into account any differences in both humans and apes' upbringing, environments, or any other factors. Subsequently, much more recent studies have thoroughly provided evidence demonstrating that apes are able to point at absent objects. Finally, the researchers list 4 possible solutions to what they describe as the pervasive superiority complex in comparative psychology research. First, cross-fostering, where apes are adopted by humans, allowing for a clear comparison between the two species, which unfortunately raises many ethical issues. Secondly, radical operationalism, in which comparisons between apes and humans are defined by variables which can be objectively measured. Third, training, which would allow for apes, like human children, to be trained in the skills that are being tested on beforehand. And finally, sampling, which in improving this by selecting more diverse human participants for studies, as well as apes from more positive backgrounds rather than sterile laboratories, would allow for less biased research. Therefore, while this study shows that research up to this point has been very biased towards humans in comparative psychology, it also demonstrates how there are many options for improving the field to more fairly compare apes and humans. Up next, Christine with more biology news.
Thanks, Caroline. Breaking news. For the first time, researchers have been able to prove that certain moths have an optimal speed at which they fly. At this speed, the moth travels in a way that is most efficient while using the least amount of energy. Biologist Carol Warfinch, along with her colleagues at Lund University, conducted a study that involved tobacco hawk moths. These moths travel at an average speed of 2 to 3 meters per second. The moths were brought into a wind tunnel where a technique called tomographic particle image biosymmetry, or TOMO-PIB for short, was used in order to determine the amount of kinetic energy exerted by these moths when they flew at a certain speed. From the experiment, researchers concluded that energy exerted from the moths flying slowly at 1 meters per second and flying quickly at 4 meters per second were generally the same. Flying slowly made it difficult for the moths to lift their wings, while flying quickly caused an increase in air resistance. The relationship between speed and power in the moths is U-shaped, in which the lowest point represents the optimal speed. From this, researchers determined that flying too quickly or too slowly was not ideal for moths in terms of conserving energy. The researchers also determined that tobacco hawk moths fly at an average rate of 2 to 3 meters per second because this moderate speed allows for the moths to fly the furthest while using the least amount of energy, allowing the moths to stay in the air for longer.
Thank you, Christine. Breaking news. Scientists have just discovered that it could be possible to turn oxyanthic cells or stomach cells into melanocytes or skin cells. Professor Carmen Levy at Aviv University and Dr. Jacob Hanna of the Weizmann Institute of Science tracked the transformation of genetically manipulated cells into melanocytes, cells that are responsible for both the production of skin pigment and is essential to the body's auditory system. Professor Levy explained that when cells develop, they differentiate into different organs with varying functions such as bone, intestine, brain, and so on. After the study, scientists have found that this process is not irreversible. It is possible to transform a cell that already has a definite role into a completely different kind. Professor Levy and Dr. Jacob Hanna discovered this by taking cells from the stomach, intestine, connective tissue, heart, and brain of a mouse. Next, they placed these cells into a solution that would activate the MITF, or microphthalmia-associated transcription factor. The MITF acts as a master regulator of melanocyte development, which provides instructions for making proteins. By taking a stomach cell and placing it into the solution, the MITF now becomes activated, transforming the former stomach cell into a melanocyte. This process has the potential to save many lives in the near future. It can be used anywhere from transplants, which could eliminate long waiting lists and cure hearing loss. And that's all we have for today. And now on to you, Amai.
Thank you. Now on to a short discovery of scientists using wild mice gut bacteria to be implanted into laboratory mice in hope of providing a better representation of human reaction. But laboratory mice have been living in sterilized, more controlled environments. The mice are not able to relate to human reaction. The gut bacteria being restored to the sterilized mouse. This will allow more realistic understanding of other bacteria and viruses that affect human life. Scientists did a research study using these new mice along with the control group sterilized mice and exposed them to a high dosage of the influenza virus. After observing the mice, the sterilized mice only had 17% of survivors, while the gut bacteria restored mice showed 92% of survivors. This new discovery of gut bacteria will allow scientists to create a complete microbiological fingerprint of microbitopia and better prepare or control later viruses affecting biology. Thank you.
Thank you, Tammy. Scientists have created a chip that reprograms cells to regenerate damaged tissue. The capability to change or reprogram cells into other types has stirred up hopes for regenerating damaged organs and limbs. But existing methods have only been tested on laboratory animals, and are shown to be hazardous and inefficient. However, a new technology could overcome these limitations. Researchers have employed it to restore injured mouse legs and claim the technique is safe enough to experiment on humans. Cells are typically reprogrammed using mixtures of DNA, RNA, and proteins. The most common process uses viruses as a delivery vehicle to provoke immune responses, but this process can turn cells cancerous. An alternative, known as bulk electroporation, exposes cells to an electric field that pokes holes in their membranes to allow genetic material and proteins in. However, this method can stress or kill them, and only a small portion is tissue nanotransfection, explained in a study published in Nature Nanotechnology in August, which involves a chip containing a variety of tiny channels that apply electric fields to individual cells. You affect only a small area of the cell surface compared with the conventional method, which interrupts the entire cell, states co-author James Lee, a chemical and biomolecular engineer at Ohio State University. Essentially, we create a tiny hole, inject DNA right into the cell, so we can control the dosage. A psychologist at Ohio State, Chandon Sen, developed a genetic mixture that quickly changed skin cells into endothelial cells, the leading component of blood vessels. Then they employed their technique on mice whose legs had been damaged by a severed artery that cut off blood supply. After 3 weeks, the legs had fully healed due to the formation of new blood vessels and blood flow increase. This is a larger model of the actual chip. By placing the device on the skin surface, the chip transforms the genetic code of the cells below, through which electrical currents flow. And these altered cells can be used to repair any organs. The transformed cells appear to secrete reprogrammed materials in extracellular vesicles, EVs, that target deeper tissue. Similar to the effectiveness as using the chip itself was injecting mice with EVs harvested from the skin of other treated mice. Neurobiologist Benedikt Berninger of Johannes Gutenberg University in Germany questions if we get EVs to convert to specific cells. While testing is only being done on pigs and mice, the team aims to begin human trials within a year. Considering what could be done, this nanotransfection process could be transformative.
Thank you, Talat. There has been a new leap in the diagnostics of autism. In previous years, scientists and doctors sequenced a patient's genome to diagnose autism. Genetic testing has advanced miles since the mid-1900s when it was first founded, yet disorders like autism are difficult to diagnose with the current genetic testing. The results reported in September by Evan Eckler suggest that the scanning a patient's entire genome could help researchers better understand all the genetic abnormalities that cause autism. These findings could eventually help doctors diagnose the disorder much easier. In 5 to 10 years, whole genome sequencing could be the most informative tool to diagnose autism, Eckler says. With a whole genome sequencing, doctors can see patients' whole genetic makeup, and in recent years, whole genome testing has become increasingly more affordable and more available. Current testing for autism looks at a broad portion of the genome for DNA insertions and deletions, when a part of the DNA has been either deleted or an extra part has been added onto it. That looked at the insertions and deletions that have previously been linked to autism, or they looked at the DNA building blocks of certain genes. However, current testing can only flag about 10 to 30% of the autism cases. Eckler and his colleagues looked at all of the genomes of patients to look for other genetic variations that might be unique to autism. Researchers at the New York Genome Center, including Robert Darnell, looked at genomes of 2,064 people in total, 516 autistic children with no family history of autism, and no genetic abnormalities detected by current testing, and the genome of children's parents and unaffected siblings. Eckler and his colleagues analyzed each family's data looking for genetic variations that occur only in children with autism. With so many families to study, the effort was a massive undertaking that required 2 million hours of computer processing time, says Tekla Turner, the postdoctorate fellow in Eckler's lab. Eckler's team identified genetic changes that disrupted genetic function
and led to different proteins productions. Genetic deletions were so small that they had gone undetected by the current genetic testing. Some of the things that were found were not genes, but were responsible for turning on the genes. Children with autism were significantly more likely to have 3 or more different kinds of genetic variations than their unaffected siblings. However, this experiment needs to be retested to prove the findings are significant enough to rely on for diagnosis. Still, the idea that multiple genetic variations could lead to autism means researchers need to dig deeper even on cases we think we've already solved, Eichler says. Whole genome sequencing could reveal additional genetic variations in children already diagnosed with the disorder. That's all the time we have for here today. Here's Hannah with more briefing.
Thank you, Alicia. Now I'm going to talk about a rare untreatable strain of gonorrhea. According to a news report from Kate Keeland released on July 6, 2017, at least 3 people throughout the world have been infected with this disease. The cell looks like this. Through sexual encounters, this number will continue to grow at an exponential rate if not properly treated. The World Health Organization estimates that on average, 78 million people worldwide are infected with gonorrhea each year. This disease specifically affects the genitals, rectum, and throat. It can lead to pelvic inflammatory disease, ectopic pregnancy, and infertility, as well as an increasing risk of getting HIV. From WE, a scientist working for the Journal of Science, PLOS, he states that from the 3 people that have been infected with this rare strain, known as a superbug, there was not an effective antibiotic. The World Health Organization contracted a study on the antibiotic resistance to gonorrhea. From 2009 to 2014, there was an increase of resistance to the first-line drug ciprofloxacin, as well as azithromycin. The last resort are treatments called extended-spectrum cephalosporins, or ESCs. According to this graph, by the year 2050, the leading cause of death will be antibiotic resistance. Therefore, it is important to find innovative ways to solve this catastrophic problem. There are currently only 3 new gonorrhea drugs that are now in development, and they are not known to be effective in final stage trials. It is important that scientists continue to create new drugs and that they will eventually be made available to everyone. That is it for today. Thank you for watching JBNN.
Talk about a rat that likes to play hide and squeak.
Thank you for tuning in. Now back to you, Rachel.
All right, Rachel.
That's probably the best one. Good.
Each other to form the patterns. He discovered a key factor to how zebrafish generate their patterns.